And can you tell me if you can see the screen or not? Are you able to see the screen? Yes or no? Okay. Everyone switch on your videos. Yes, students, switch on your videos quickly. Jesslyn, Muniba, switch on your videos. So, in the previous class, we saw how basically the biotechnology was used in the field of medicine as well, just like how it was used in the field of agriculture, right? It was used in the field of pest control. Now we will be seeing how biotechnology or the technology of recombinant DNA is used in the field of therapeutics or medicine making. So in the previous class, how we have seen that out of the different medicines that are sold, 30 RDT therapeutics are approved and 12 of them are being marketed in India as of now. Now, some of the drugs that we saw as a list, human insulin, there was streptokinase, then there was erythropoietin, hepatitis B vaccine, human growth hormone as well. So furthermore, today we're going to see how the recombinant insulin was made. Okay. Now, can you tell me, students, which particular gland or organ is responsible for the formation of insulin?
Thyroid?
Hmm? No, not thyroid. It's a gland that is a mixed gland. It is endocrine also, exocrine also.
Pancreas.
Which one?
Pancreas.
Yes. Pancreas. So pancreas is that gland in our body which when it acts like an endocrine gland, it synthesizes the insulin hormone. And who knows the function of insulin hormone? Insulin hormone is important for the glucose metabolism in our body, right? Meaning insulin lowers the level of glucose in our body and the opposite of it is glucagon, which increases the level of glucagon in our body. So when we talk about the recombinant insulin and how it is made, so basically pancreas, they have two types of cells present in them, alpha cells and beta cells. Alpha cells of the pancreas, they synthesize the glucagon hormone, whereas beta cells of the pancreas, they synthesize insulin hormone. Both of these hormones are involved in the glucose metabolism and both of them act against each other. Okay, so we can also mention one more point that they are antagonistic to each other. So these two hormones, they are antagonistic to each other, meaning they act against each other. Understood? Like if glucagon increases the level of sugar in our blood, insulin lowers the level of sugar in our blood. So that is how they work against each other. But both of them are involved in controlling or regulating the level of glucose, that is sugar, in our body or in our blood. Now, when this insulin is not secreted in a sufficient amount, when the deficiency of insulin takes place, so a disease that is called as diabetes mellitus happens, right? So you might have seen, if you know people that have diabetes, you might see that they take insulin shots. They inject themselves with the insulin hormone before having a meal. Have you people seen that? Why? Because their body or their pancreas, they're not able to make the insulin hormone, which is very important. If a person doesn't have insulin, because of the spike in the sugar, a person can have so many different disorders. A person can even die. So earlier, what used to happen to treat these people, to provide them with the insulin was taken, or you can say it was sort of extracted from the pancreas of the slaughtered pigs and cattle, meaning slaughtered cows and slaughtered pigs. These two things from the slaughterhouse, people used to go there, and because their, you can say, genetics or their genotypes are very like similar to human beings. Okay, so the insulin was taken from their pancreas, and then it was, like created for human beings or for human use. But at that point of time, this created a lot of problem, because first of all, the first thing is that since the insulin was taken from animals, which are like completely different species, so it created a lot of allergic reaction to the people that were taking it. Second thing, many people were not ready to take these insulin. Why? Because you can say it's unethical to take it from animals. And other than that, different people have their different religious beliefs. For that also, people were not comfortable with it. So then this whole thing had to find some sort of alternative. So why does the insulin, this question. Can you tell me, you people only tell me, I asked you that if you have seen people injecting themselves with the insulin, right? Can you tell me why do people not take the pills, like the medicine pills, capsules or stuff of insulin rather than they have to inject themselves?
When you take pills, it takes more time for the
It takes more
enzyme
time.
uptake. Like it takes more time, I don't know.
That's actually true, but not in this case. Let's just not say time is not of the essence. Why can't you take insulin orally? Why do you have to inject it? Think the answer is very easy.
So
Think
that
a
it
little.
can directly mix with the blood.
That's true. Through the injection, it directly goes into the bloodstream. That's right. Then what is like when we eat something that also goes into the bloodstream? It takes a little time, but it also goes into the bloodstream. And why, I mean, not taking it through the oral injection? Why do we have to inject ourselves? Achha, I'll tell you. When we take anything into our alimentary canal, into our digestive tract, the thing is that our digestive tract, before absorbing something, it basically kills that particular thing. It digests it, right? Now, if the insulin goes into the alimentary canal orally, the composition of the insulin, the structure of the insulin is going to get damaged, because in alimentary canal, it will anyway get damaged, right? So oral administration can degrade the insulin. That is why we take it via injection through directly into the bloodstream. Other than that, the reason that you people said that through the bloodstream it is quicker, that's correct. That's correct too. Okay. Now, in humans, the insulin is made up of 3 chains. Okay. Like we are going to the structure level of the insulin. So A, B, and C. 3 chains of insulin are there. And insulin is, whenever it is synthesized as a prohormone. Prohormone means it is inactivated initially, but it has to be made active. So when it is a prohormone, it only has like basically A, B, C chains are there, and this is an inactive insulin. To make it active, to make it mature, the C chain has to be removed. Okay, to make the insulin mature. But now, since we are doing it through the recombinant DNA technology, so what do? We make the chain A separately, we make the chain B separately, and then they were joined together using disulfide bond. So there's no question of C chain here. There's no, you can say, immature insulin made. See, this is the technique. Basically, you from the pancreatic cell of human, you take out the insulin gene. Okay. Then You take out the plasmid from the bacteria. You cut the insulin gene and you cut the bacteria with the same restriction enzyme. Then, why are we cutting it with the same restriction enzyme, students? Why not different? Why not the ones that we want to cut it with? So this too you should know by now. Why are we cutting it with the same? Why is it important to cut it with the same restriction enzyme? Why? No one. Eh? No one. See, the reason why we have to cut it with the same restriction enzyme is because we have to join them, because cutting them with the same restriction enzyme would create the same kind of end so that they could be joined together. Sticky ends will be created. So once you've cut them, now you join them with the DNA ligase. So the plasmid along with the gene of interest you've incorporated, and that is your recombinant DNA. Now you take this recombinant DNA and introduce it into the host cell, and whenever the host cell will multiply, this insulin gene will also multiply, right? And then you're going to extract it out in a medium, like grow it on a larger scale and then extract it out. And what you have at the end is the recombinant insulin that is to be provided to the patient. So here, this is for chain, that you do this for chain A and chain B, right? And then you combine them together. So chains A and B were produced separately. They were extracted and then they were combined by the disulfide bonds to form the actual human insulin. So in 1983, Eli Lilly, very important. This is so important for MCQ, for true and false. So this is an American company that prepared the two DNA sequences, A and B of the insulin. And they were successful in doing so, and thereby the human insulin was synthesized. So this human insulin is also called as humulin. It is called as humulin. So human insulin, like human plus insulin is humulin. Clear? Any doubts, students? Okay. Now we are seeing the usage of biotechnology in the therapeutics, in the medicine. So there is one more thing, which is a corrective measure that is called as gene therapy. So now, basically, this is the name given to the methods that aim to cure an inherited disease by providing the patient with the correct copy of the defective gene. Now, you people know that because of the mutation, there are several diseases that can be caused. Now, those diseases can be treated if you know the gene that is being altered and you give the right form of that gene. So gene therapy has now been extended to include attempts to cure any disease by introduction of a clone gene into the patient. Meaning the corrective gene, if you introduce that into the patient, you can treat the genetic or gene disorders. For example, gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. Here, genes are inserted into a person's cell and tissues to treat a disease. So here, instead of giving medicine, you're giving genes to correct it. Correction of a genetic defect, it involves the delivery of a normal gene into the individual or embryo to take over the function and compensate for the non-functional gene. Is it clear? Like basically, if a person has any kind, like there is a gene that is defected, okay, defective in that person. So you take the correct form of that gene, which you might have developed recombinant by the recombinant DNA, and then you introduce it into that person that has the disease, and that person is treated. But there are things that go behind this also. For example, this actually has been done as well. So the first clinical therapy of the gene was given in the year 1990. This is also very important. It was given in the year 1990 to a 4-year-old girl that had adenosine deaminase deficiency, ADA deficiency. So basically, this enzyme is crucial for the immune system to function because in its absence, the lymphocyte proliferation is inhibited. Now, you people know lymphocytes are those cells that provide immunity, right? So, when a person has ADA deficiency, so they have, you can say, lymphocytes do not proliferate, meaning they do not divide and become several in number. Okay, so without the T cells, the ADA deficient children, they are wide open to the attacks of viruses and different kinds of, you can say, diseases, right? And SCID, there is a disease called as severe combined immunodeficiency disorder that is caused due to the deletion of the gene for adenosine deaminase. So now in some children, the ADA deficiency can be treated by bone marrow transplant. If you transplant the bone marrow, because you know that lymphocytes, they proliferate, like they are made in the bone marrow and they mature in the thymus, the T lymphocytes, isn't it? And B lymphocytes made in the bone marrow, mature in the bone marrow. Remember, human health and disease, forgotten. Remember? So, it can be treated by enzyme replacement therapy. So, patient is given the intravenous. These are the methods of convention treatment, like not RDT. These are like what all things can be done. First, bone marrow transplant can be done. Enzyme therapy can be done. These things can be done. But these are not completely curative, meaning it has to be periodically done. Okay, like a person has to constantly go to the doctor to get this thing done in them. Then comes the gene therapy with the usage of RDT. So here, the lymphocytes are taken from the patient. That the patient that has ADA deficiency, their lymphocytes are taken out and they are grown outside the body. Like in a laboratory condition, they are grown in vitro. So a functional adenosine deaminase cDNA. cDNA is a circular DNA or a complement DNA that is using a retroviral vector is introduced into the lymphocyte. So now the defective lymphocytes were taken out. Okay, the enzyme which was lacking, that enzyme was put into the lymphocytes. And then these lymphocytes were sent back into the patient's body. And obviously, you know that these cells are not, you can say, immortal. They are mortal. So once their lifespan is over, they die. So a person has to regularly go to the lab or wherever, and they have to be injected with the RDT cells. But if this treatment that we are talking about, the adenosine deaminase treatment, the gene therapy is done during the embryonic stage, while, like, for example, because there are many tests during, you can say, that is done during pregnancy to detect any kind of abnormality in the fetus. So if, like, let's just say this is detected. So what can happen? If at that point of time only it is done, the ADA treatment is done, then it would be a permanent cure. So gene therapy is clear to everyone. Do you people have any doubts? Yes or no? Also keep your videos on, students. I'll let you switch it off when you write, but right now while I'm teaching, keep it on. So this much is clear to everyone. Does anyone have any doubts in the gene therapy? No. Okay. Sure, no one has any doubts? Now coming on to molecular diagnosis. Now diagnosis means to find out whether which type of pathogen is there in the body, which type of disease nature is there. So to find out these things, we call it as diagnosis. So diagnosis is basically a procedure of determining the nature and the cause of a disease. So during the time of disease, what happens is that when do we think that we have a disease? Can you tell me? Like, when do you people think that you are suffering from some sort of disease? What happens to you that makes you think that you have a disease?
Certain kind of symptoms.
Yeah, symptoms appear, right? Some sort of symptoms appear. You have fever, you have cough, and some way or the other, like you have body ache. Different diseases have different symptoms, right? So by the time the symptoms are shown in your body, the pathogen because of which the disease is being caused, that pathogen has increased a lot by that time, like thousands and thousands of number. But when we talk about molecular diagnosis, so molecular diagnosis is the one wherein you detect the presence of pathogen before they have multiplied a lot. So during the time of disease, the presence of pathogens such as bacteria, viruses, worm is detected whenever, like you people only told me, symptoms are produced, right? Any kind of symptoms. But when you produce symptoms, it means that during that time, that particular pathogen has increased a lot. So let us see diagnosis method. The traditional method, the basic method is blood test. Right? The blood test, if there is a high WBC count, low platelets, like that, you can find out whether or not a person has any disease. So there's blood analysis, there's serum analysis, urine and stool samples, they are taken for testing. So in this, early detection is not possible. Early detection means much before the pathogen has increased. These things are detected only when the pathogen has increased a lot and you have started to show symptoms. Whereas the different methods of modern molecular, like modern diagnosis, are the use of RDT, the use of PCR, that is polymerase chain reaction, the use of ELISA, that is enzyme linked immunosorbent assay. So in the modern method of diagnosis, early detection is possible much before the pathogen has spread, widespread. So let us see, the first is the usage of PCR, that is polymerase chain reaction. You people remember the PCR steps? Denaturation, annealing, extension, those things you remember? Yes or no? What was PCR used for? Even if you give me one word answer, I'll be happy. The PCR is used for?
HIV.
Huh?
HIV.
HIV. No, no. For what purpose? For what? Like what? HIV ka kya? Like what of that thing? For what purpose is that PCR used for? In general, not just for disease related, in general.
To make many copies
Mm.
of the same
To
thing.
make several copies, right? Which is called as amplification. That's the basic principle of PCR, right? You make thousands and billions of copies. So PCR is used for the detection of like even very low concentration of bacteria or virus much before the symptoms are produced. How? By amplifying their nucleic acid, meaning either DNA, you can amplify that, or you can amplify their RNA. Now, PCR is now used to detect HIV in suspected AIDS patients. PCR can be used to detect mutations in the genes in cancer patient and many genetic disorders. Meaning, if a person suspects that that person might have a disease, so their extract can be taken. It can PCR be done and then be confirmed that yes, this particular pathogen is there much before the symptoms are produced. So this is one method. The second method is ELISA, that is enzyme-linked immunosorbent assay. So this is antigen-antibody interaction-based technique. So infections by pathogen, they can be detected by the presence of antigen or antibody synthesized. Like antibody, our body is going to make the antibody, and antigen is that foreign particle. So if that can be detected, then a person would know that, okay, this is the particular disease is going to be caused. So that is enzyme-linked immunosorbent assay, which is based on antigen and antibody interaction. Everyone knows what's the difference between antigen and antibody? Is there anyone who doesn't know it? Everyone knows it? Okay, good. Now coming on to transgenic animals. Now transgenic animals are those organisms that have their genetic, or you can say genes. They have their genes modified. Their genes are not normal as to how they were born with. Their genes are modified with the usage of biotechnology or recombinant DNA technology. So, these are the animals that have gene or DNA manipulated to possess either an extra gene or remove some of their genes. Some of the examples of transgenic animals are rats, rabbits, pigs, then sheep, cows, monkey, fishes. These are some of the examples of transgenic animals. Out of all of them, majority of the transgenic animals are mice. And you people might know that most of the drug testing and cosmetics testing, any chemical testing, it's mostly done on mice. Right. Sometimes it's done on rabbits also. But that's it. Now, why do we produce transgenic animals? What is the need? Why are we troubling them? Why are we torturing them? What's the need? So some transgenic animals, they are produced for specific economic trade. Meaning you may need something. For example, like cows. Cows are there. Cows provide milk. But sometimes it may be there. Cows are providing milk with less fat content. So maybe you modified the genes of that cow to produce milk with higher fat content, right? So now you're getting an economic trait out of them. Second, other transgenic animals, they are produced as disease models so that any kind of drug testing, any kind of disease, like for example, there are so many bacterial and viral pathogens available that cause diseases in humans. Now, when like you, for example, you are doing some sort of research, you want to study about a particular pathogen and how they infect humans and what they cause. Will you go and take that pathogen and inject it into your friend? Like, oh, I'm sorry, I wanted just to study the symptoms. Can I see your symptoms? No, right? You'll go to jail for that, isn't it? But the same thing is done. Many transgenic animals are developed so that you can provide or you can take that pathogen, put it in those animals. It's very cruel, but this is what's done. So you take it into those animals and then you study the disease through them because you can't do it for humans. Because apparently human beings have coded themselves as to be the best and superior of all, and then everything else below them. So that's the case. Now, benefits from transgenic animals are under three broad categories. Medicine. Cows that produce human protein rich milk. See, human milk is very, very high in antibodies. Everyone knows that, right? Cow's milk is not that high. But what if you take the gene of the human of producing a very high protein rich milk, put it into the cow, and now the cow produces the milk that has high protein content, just like human's milk. Then there's agriculture. The sheep that grows more wool. So there will be more wool in the market. There will be better economic value of the woolen clothes and everything. And then industry. Goats that produce silk. Imagine. Silkworms are there, right? And do you people know that silkworms, if you, I don't know the like for getting few yarns of silk, you have to kill so many silkworms. So when you can do that recombinant, like using the RTE so that you can create it in a mass number, so for that it is utilized. Under the medicine category, the transgenic animals can be used for studying the normal physiology and development, to study diseases, and it is used to produce biological products as well. For example, alpha antitrypsin for the treatment of emphysema disease. In the year 1997, not too long ago, very recent only, the first transgenic cow was made whose name was Rosie. So she was the cow that had superior protein content in her milk. Then there was vaccine safety testing also, like mice are used for vaccine safety. Chemical safety also, like all those products that we use on our face, they are tested elsewhere. And under the agricultural category, The transgenic animals, they are used for breeding, they are used for quality checking, they are used for disease resistance checking as well. This much is clear. Any doubts, students? This chapter though is easy, na? It's like story. But see, the first biotechnology chapter, because of all of its like technical aspects, the whole entire recombinant DNA creation, that part you should know very nicely. Okay. So once this chapter gets completed, you start with the biotechnology principle and processes and learn it very nicely. Understood? Now coming on to the ethical issues. Now, obviously, if you're playing with life, if you're creating something for human beings, if you're creating something out of animals, so there are a lot of things, there are a lot of ethical issues that goes behind it. So that comes under bioethics. What is ethics? It is a set of standards by which a community regulates its behavior and decides whether something is legal or illegal, legitimate or illegitimate. Bioethics, they are a set of standards that regulate the activities in the biological world. Obviously, if you're using animals, then you have to follow certain rules. You do not have to be cruel. So there is this organization called as Genetic Engineering Approval Committee. So this organization, it is set up by the Indian government, which makes important decisions regarding the validity of the genetically modification research. Like, not, see, you can't just think of anything and you do it. That's not possible. You have to get it approved by the government, then only you can. Understood? So now coming on to the topic of patent. What is a patent? How many of you know what a patent is? Have you heard these things before? Patent, trademark, copyright. Heard it before? No.
Ma'am, not
Yes.
patent.
Hmm?
Not patent.
Patent you've heard?
No.
No or yes?
No.
No. See, patent is like, for example, anything can be patented. For example, you have an idea about a research. Okay. You have an idea about any research. If you get that idea patented, and if I try to copy you, I will get fined for that. Because now your idea is legitimately set like that. And I will get a fine because I'm trying to copy you. You might have seen that like there are big chains of food, like there are so many like KFC, Domino's, Pizza Hut, all of these are there. So when the same exact like places for junk food are open in the city, they cannot use that name. Have you seen that? Like those local, like for example, KFC sells burgers. So there is no local company that can take KFC name. It's not even possible. They can't copy the name. No other company can copy the name of the thing that has been patented. That is a brand name or that is a trademark. So that is what a patent is. A patent is a right which is offered by the national government to the inventor. Okay. For a limited period of time in the exchange for a public disclosure of an invention. And basically, when you're patenting something, if anyone else uses it, they have to use your permission. For example, now, like whenever you people might know about YouTube, right? So on YouTube, there are people that upload videos. If you watch blogs and stuff, you might have seen that they don't put any like movie music in their videos. They can't. Any movie music they can't put, any singer's song, like any kind of singer, whether it is Indian singers or international singers, they can't use their songs for more than I think 10 seconds. For more than 10 seconds it's not allowed to use because they'll get a copyright on that. So it's like they'll get fined for it or their videos will be taken down because that is a right reserved for the original inventor of that particular thing. So when we talk about the patent, so it is a right which was given by the national government. Now novelty, and what are the things that the patents are supposed to satisfy? Novelty, non-nomination, and utility. Acha, Jobita and Aisha, why are you people so late today? You people are almost like half an hour late to the class.
Mom, I had connection problems.
Hmm? I can't
I
hear you.
had internet problem.
Internet problem. And Jobita, what about you? Half an hour late?
Ma'am, today I got up late and I just ate my lunch right now, so that's why I got late.
Okay. Okay, so when we talk about novelty, it implies that the innovation must be new. It should be something that has not been done before, right? You cannot copy someone else's thing and be like, no, this is my idea, I'll get it patented. No, it's not possible. You have to create something that is new and unique to you. Okay, so a patent, it is granted for an invention. Okay, meaning maybe you have invented some product. It is granted for an improvement in an earlier invention. Some improvement you can do, like some new things you can add, right? The process of generating a product, the process, like the way that you have created, like there are so many things available, like cookie recipe, let's just say. So there are people, like cookie recipes are everywhere, right? But now let's just say you have created a new recipe wherein you have shown different method. You have added some new ingredients. So now that is your patent. You can get it patented. No one can copy that. No one can call it theirs. If it is originally yours, no one can copy it. So that is a concept or a design. So initially patents were granted for industrial inventions only, but now it is granted for many, many things. But at present, it is being granted for biological entities and for products that are derived from them, and we call them as biopatent. So, for example, I'll tell you, rice is a very important food grain in India, right? In many states of India, it is a staple food. And you know that there are so many different varieties of rice in India, like 2 lakh varieties of rice. Imagine so many states, so many areas, and every area has their own unique variety of rice. The rice diversity of India is richest. Everyone has had basmati rice, right, for biryani and stuff. So those basmati rice, they are unique in having a very unique aroma and flavor, and they are the most superior kind of rice. Imagine there are 27 varieties of basmati rice that are being grown in India. Now, if let's just say there is any other country other than India that tries to create basmati rice, they can't get it. Like they can't say that it is their idea. They can't say that it is their invention because it is already being marketed and present in India ever since time immemorial, right? It has been there for centuries and centuries and centuries, right? Now, associated with this comes biopiracy. Biopiracy, basically the use of bioresources by multinational companies without proper authorization, meaning you have used someone else's idea, you've taken someone else's concept, and you're not giving them credit, you're not providing them the payment also, right? I'm using your idea, so here's your payment for that. No. So that term is called as biopiracy. So most of the industrialized nations, meaning those bigger countries and better developed countries as compared to India, they are rich financially, right? They have a lot of money as compared to India, but they do not have resources. They do not have traditional knowledge as compared to India. They don't even have the biodiversity. So traditional resources, it includes all those organisms that can be used to derive commercial benefits. Traditional knowledge related to bioresources is the knowledge developed by various communities, right? Like for example, in India, before these antibiotics and modern medicines were developed, people used to extract medicine from the plants and use it. And it worked fine for them because they had that traditional knowledge. They knew what is going to work, what's not going to work. Now the same exact thing, the same exact plant is being it's being bought from them, and then chemically, or you can say on a larger scale, a particular medicine is being produced and then marketed. So see how people that have, that have better financial opportunities, they use the traditional knowledge of other countries and derive benefits from that. So that is why Indian Parliament has recently cleared the Second Amendment of the Indian Patents Bill that takes such issues into consideration. About the patents, biopiracy, all of that comes under this Indian Patents Bill. Clear, students? So with this, our chapter is completed. Finally, it is completed. Do you people have any doubts up until now? Anything that you have not understood? Everyone has understood everything? Okay, Acha, tell me which is the next chapter that we shall be starting next? Any requests? No. Please stop teaching.
Evolution.
Evolution. You want me to start evolution? Okay, I'll start evolution next. Okay, what is the last thing people?
Is it okay if we do revision? Small
Revision.
revision. Yeah.
Revision of what?
Molecular basis.
Molecular basis of inheritance. Where you have an
Yes,
exam.
ma'am. Yeah.
When?
Second week of September.
Second week of September. Okay. So you can come, like you revise that chapter, you learn that chapter, you come. We'll just go through it. Okay. I won't be able to revise it properly because there are many chapters left to complete for you people. So if you like have any particular doubt in any topic, I can teach you that. Understood? But from the next class, being from tomorrow, I will be starting evolution. Okay. All
Okay,
right.
ma'am.
Tell me, students, what is the last thing you people wrote down? This chapter is clear to everyone, na? I hope you have no doubts. Okay.
No, ma'am.
What is the last thing you people wrote?
Plants that make their own insecticides.
Plants that make their own insecticides. Which line here? Endotoxin part is done? No. Part before this? Or you people right now?
Done, ma'am.
Everyone's done. Shall I scroll down?
Done.
Everyone's done, students? Students, are you done? Everyone's done?
Yes, ma'am.
Done. Then done.
And Done, ma'am.
And 7, ma'am.
Done. Done, ma'am.
Students, you right. I'm just coming in 1 minute, okay?
Yes, ma'am.
You done, students?
No, ma'am. Done.
Then can you please scroll down?